Okay, good morning, folks. Happy Tuesday. So today we're going to be talking about antimicrobials. So this is, I think I mentioned last day, this is an area special to my heart because I spent a lot of time, a lot of years thinking about penicillin and how that works. And so we'll definitely get into that. Before we get into today's topic, I just want to spend a few minutes talking about the midterm. If there's some time at the end, I will come back to this. But mostly I wanted to just kind of go over what to expect for midterm number 2. You can see we've got, when I'm looking for some inspiration, I'm always looking at Calvin and Hobbes. And you can see that Hobbes has said, have you studied for microbiology yet? And Calvin says, no, I'm waiting for inspiration. You can't just turn this kind of thing on. You have to be in the right mood. And he says, what mood is that? And Calvin says, last minute panic. So hopefully nobody is going to take that strategy, but we are a week away. So now is the time to at least worry a little bit and think about studying. If you haven't started already, now would be a good time to start to think about studying. So in terms of what to expect, it's going to be very similar to the first midterm in terms of format. The content will be different. I'll talk about that in a moment. I'm just trying to get my slide advanced. For some reason it's not. Oh, there we go. So just like before, we're going to be using the lockdown browser. Just like last time, I believe I do have a practice midterm on Moodle that you can take a look at. And I don't know if you're asking for a practice one online. If that's the case, I do not have one set up. If you're concerned about internet connection issues, I know that there were people previously had some, internet troubles. You can probably find you a place on campus. That's the kind of thing you should let me know. Check with the library. I know the library has rooms to book, and then they have open areas too that can be used for this kind of purpose. So if you do have any concerns, you have a week to figure it out. So I think somebody's asking if there is a practice midterm on Moodle. No, I don't have a practice midterm on Moodle. I don't have the online one on Moodle. I just have a paper type one up on Moodle that you should be able to find. So what's the format going to look like? So pretty similar to the last one. This isn't necessarily finalized. This is just kind of my plan. I'll probably stick with it for the most part. I can't even remember the last one now. I think maybe the last one had slightly less written. Maybe the last one had 10 marks written. This one will have 15 marks written because I'll have a fill in the blank. I'll have a short answer around disinfection or sterilization or something like that, and I'll have some sort of case study. And then everything else will be like basically what you saw before, some sort of combination of multiple choice, matching, and true and false. So someone says, in the last midterm, some questions like the HIV one did not appear on mine. Are you going to choose a case study? I have not decided yet since I haven't written the exam. So my goal is to have 1 or 2 case studies and either give people a choice or I'll just pick one. I haven't decided yet. So like I said, I got to spend a little bit of time in the next few days kind of thinking about this and coming up with some questions and seeing what I've done previously. But like I said, the format should be nothing new. So just a reminder for the written questions that you are thorough and give examples where necessary and those kind of things, particularly longer answer questions like the disinfection sterilization one, right? It's worth 5 marks. So you don't want to give me a 2-mark answer, right? If this was written on paper, I usually tell people that 5 marks is somewhere around half a page or more. Kind of depending on the size of your handwriting and all that. Typed, that's about 5 sentences. If you're not using sentences point form, that's fine. But a sentence isn't just something short, right? A sentence is more of a complete thought. So if you're telling me something about ethanol, right? Don't just say ethanol is the best answer. Okay, and why, right? You don't want to expand on those kind of things. So you can see here I've written that it's going to be topics 7 to 10. So Some people were asking, I think, a couple of weeks ago whether it is a shorter midterm. No, it's not. These topics here were all about 2 lectures each, so we've covered quite a bit of material. And this is why we have our second midterm now, because I don't want to cram too much into a midterm. I want to kind of have it just right. So topic 7 to 10. So we haven't covered the antimicrobials today. I am hoping to today get through about 2/3 of that topic, would be my guess, probably the antibacterial part. And then the rest will be covered on Thursday. So let's get into the antimicrobials. Like I said, if I have a little bit of time at the end, I'll talk about the midterm a bit more. And hopefully a little bit of time on Thursday to do some review questions. And but just a reminder, like I said, get started studying if you haven't done so already. There's a little bit more technical stuff in these topics, a little bit more strange vocabulary, and antimicrobials, there's a lot of weird words too. Okay, so topic 10, antimicrobials. So topic 9 was kind of about controlling microbes, in the environment, right? Sterilization, disinfectant, antimicrobials are inside the body, right? So kind of a different aspect of these kind of things. So we're not going to be drinking or injecting any disinfectants, I hope, but antimicrobials, meaning the drugs, we might be doing that. Okay, so here's a good place to start. We're going to start off with history. And this is a little thing that popped up on the internet a number of years ago. It's pretty cute. So I'll share it with you. History of antibiotics, 2000 BC, here, eat this root. 1000 AD, that root is heathen, say a prayer. 1850 AD, the prayer is superstition, here, this potion. 1920 AD, that potion is snake oil, here, swallow this pill. That pill is ineffective. Take penicillin, 1945. 1955, oh, the bug mutated, try tetracycline. 1960 to 1999, 39 more, oops, here, take this more powerful antibiotic. And then year 2000, the bugs have won, here, eat this root. So looks like this is about 20 years old, whoever wrote this, anonymous, but it's kind of cute, right? Talking about we didn't know what to do, then we found penicillin, and penicillin wasn't effective, and so we have other options, and now there's a lot of resistance, so what are we going to do about that? So some quick definitions for you, and we'll probably see a lot more definitions as we go along. Pharmacology is the study of drug action. So I think next fall you take a pharmacology class. And so that obviously includes antimicrobials, chemotherapeutic agent or chemotherapy. Often people think cancer when they think of chemotherapy, but it can apply to pretty much any drug that acts against a particular disease. I went to a chemotherapy conference years ago, and it was mostly antimicrobials. Because people are thinking about, hey, we want to treat this disease. It's just sort of in the common speech, kind of stuck with cancer, right? Antibiotic. So you can see antibiotic there, I have written down as something that can treat an infection, right? But usually when people say antibiotic, they are talking about antibacterials. So why is that? Well, it's just kind of common speech. We have way more antibacterial drugs than all the other ones combined. So often when people are taking an antibiotic, they are taking an antibacterial. And like I said, commonly people, when they say antibiotic, they really are referring to an antibacterial. Other ones were usually more specific. We might be saying we're taking an antifungal or an antiviral or something like that. So I'm going to try to not use the word antibiotic unless I'm in them all as kind of a big picture kind of thing. I'm going to try to say antibacterial or antiviral to be specific. So I do have a little bit of history for you. There's some points to be made along the way about these drugs. If you look at what was going on more than 100 years ago, we did not have penicillin. We did not have really a lot of treatments for a lot of things. And you can look at the stat. I've seen the stat in a number of places, and I've actually seen higher numbers from other sources, but this was from the CDC, so I figured it's probably relatively reliable, and is saying about 1/3 of children were dying before the age of 5 due to infectious diseases. And you can imagine all the possible infectious diseases that us or our children have had, and how easy it is nowadays to go in and get some sort of treatment because, well, we have things like antibiotics. Back then, that was not the case. Either your immune system cleared it or it did not. What could you do? You could maybe treat the symptoms. You could make the person comfortable. You could pray. Those were kind of, the things that people were doing. There were, of course, all sorts of, folk remedies and weird things people were doing, and all sorts of variety of cultures. Even today, there's still weird things people will do. But the reality is it was a pretty high mortality rate. And so if you take a look at the past 100 years at just the death rates in populations, you can see it has plummeted. It's really quite incredible. You can see around 1900, so about 900 deaths per 100,000. And you can see all of these momentous events, right? We've got things like water sanitation in there. We've got 1918, there's a spike, right, due to the Spanish flu, right? And then it just keeps going down with immunizations and drugs and those kind of things. So really, just very, very incredible to think about all of those things that have happened in the last 100 years. If you know any old people, right, some grandparents or anything like that, you can ask them if they remember any of these events, right? My mom, in particular, I remember talking to her about the polio vaccine, and really fascinating for her to have lived through that and to tell me her experiences of remembering these polio epidemics. And an uncle of hers was paralyzed, and et cetera. Some really fascinating things. Somebody's asking, do we need to know these dates? Nope, don't need to know the dates. Another little graph I found as well online, and you can see how much things have changed in 100 years, right? 10 leading causes of death. Obviously, this is for Americans, but Canadians are probably relatively similar, or it could be worldwide, I'm not sure. But you can see 1900, right? Top 3 causes of death are pneumonia, tuberculosis, diarrhea. Diphtheria is in the top 10 as well, right? Year 2000, you can see, pneumonia is still there. It's dropped relatively significantly, and diphtheria is gone, tuberculosis is gone. Not that tuberculosis is not important, it's actually very important in some countries. But in Canada, most of us may not even know someone who's ever had a case. I think I know one person who had a case in my lifetime. I've met personally. It was some kid when I was like in grade 9. He missed half a year of school due to it. And so everyone knew he had tuberculosis. But besides that, I can't think of anyone else I've known who's had tuberculosis. So all these changes, a lot of it to do with drugs and vaccines. So let's talk about drugs, okay? So you can imagine in the past 100 years, and all the types of things that have happened in terms of what we've understood about chemistry and those kind of things. And so along came this guy here, Paul Ehrlich, and he was a chemist, and he kind of had this concept of what we call the magic bullet. So this idea that, hey, maybe we could find a chemical that will kill a pathogen and not kill the human, right? So he starts going through these chemical libraries and testing these drugs, and he was testing it on syphilis. Syphilis, before any antibiotics, was a very significant disease because it was not treatable, and a lot of people had it. Sexually transmitted disease, completely untreatable. Eventually, it has very devastating chronic effects, can infect your brain eventually. So he came up with this compound, 606, and it turned out it was a cure for syphilis. So this was really quite remarkable. So it has a name, Salvarsan, and it started to get marketed, and lots of people were getting treatment. So here's a treatment kit I found on the internet from a science museum, and really basically what they were doing was spreading the chemical over a person's genital area. A person would have to go in for several treatments, and they would, these doctors who were treating them actually got quite wealthy from this because it was several treatments. It wasn't like the most effective antibiotic, but it was an antibiotic. It was an antibacterial compound that had a specific use. So a cure for something that never had a cure before. Really, in some ways, Magic, right? So after that, people started, they had this idea in their head, hey, we can find cures. We can cure bacterial infections. So a little bit of research put into this. I mean, we're not talking about nowadays where billions of dollars are put into research. We're mostly talking about people and companies, and they've got a little bit of a side project kind of going on. And sometimes people got lucky, and people had this idea in the back of their mind that maybe we could use chemicals to treat things. So another one had come along in 1935. You can see this is Prontosil. This cures streptococcal infections of different types. So group B strep, for example, is the one we're talking about that can get spread from mother to baby. And it's called childbed fever or puerperal fever. And so there's another one there. So again, this one's not the most amazing drug, but again, it is a cure where there was no cure before. So let's talk about penicillin, because this is really where antibiotics started to come into their own, being really amazing, powerful treatments for things. So this is about 1928, and Alexander Fleming was a Scottish doctor. He was working in London, and he was getting rich treating syphilis patients with salvarsan. And so he's got this idea. He's thinking about antimicrobials, and he's doing a little research on the side. He's got a little bit of money. You can see there he is with a bunch of petri dishes. Who knows what he's doing there? And he had a little experiment, and he figured he was going to grow some cultures, and I can't remember, it was a long weekend or some sort of weekend, and I think he was going bird watching or hunting or something like that. So rather than putting his dishes in the incubator, he wanted to just grow them a little slower, so they'd be ready on Monday. He just leaves them on the bench, and one of them got an infection. So here is probably what it looked like, something like this. We don't have a photograph of his original plate. But you can see he was trying to grow Staphylococcus, and he comes back and it's got a mold infection, right? And the mold has a name. It's called Penicillium. And you can see around the mold, there's this clearing. So this is the kind of thing that happens once in a while, right? You work in a microbiology lab, your plates get infected, often you just throw them out. But he made this observation. He's like, the bacteria are being killed. This is very significant. So he decided to investigate a little bit more. You can see there's a little micrograph there of what the mold looks like, Penicillium. It's actually quite a beautiful structure. And so he's like, okay, he took this mold preparation and he made what he called mold juice initially and tested it against a bunch of different things, right? And so, hey, he's in a microbiology lab. What does he have? So he tested, Staphylococci, pneumococci. So that means streptococcus, other streptococcus, gonococci, meningococci. So all those are gram-positives, by the way, if you didn't notice. And he found it was useless against E. coli, useless against TB and typhoid fever. That's typhoid fever is caused by salmonella. My big question is why he didn't test syphilis. Maybe he was not able to grow it in the lab at the time. I'm not sure of the history behind that. But wow, right? I mean, these are some pretty significant infections that were up until this point relatively untreatable. He did some further investigations. He was not a chemist, so he had a hard time, isolating the compound. He did give it a name. He called it penicillin. He even injected into rabbit and mouse, no apparent side effects, and even treated a couple of patients by kind of washing the mold juice over an infected eye and an infected leg, and he had some great success. Now you can see this last part here, right? His preparations quickly lost their potency. So it turns out penicillin, it has got to be super fresh, like you've had to have extracted it in the last day. And so this is discouraging, right? You can't have something just sitting on the on the shelf and hoping to use it. And so he started to get into other things, and eventually kind of just went on to other projects where he thought, okay, I'm going to study a lysozyme. That's an enzyme he found in his nose that also had antibacterial properties. Like I said, he kind of moved on. He published, and anybody who said, hey, can I have some of that mold? Because this is pretty cool. And so he shared it with tons of people. So life went on, 1929, and you know, lots of people got this mold sample, and lots of people were very interested. And along came these guys here. So Chain was a student in Florey's lab, and he requested the mold. And these guys were chemists. And they isolated the compound, and they found out if you dry it down, like you lyophilize it, freeze dry it, whatever word you want to use, you could preserve it, right? It didn't lose its potency so quick. And they said, hey, let's take another look at this thing, because I think it's got some potential. So they did some experiments where they you know, took some mice and they injected some streptococci into the mice, and the mice died. Okay, so not a problem. Unless you're the mouse. And then they did a second experiment where they injected the streptococci and a bunch of penicillin, and the mice lived. And they're like, wow, this is you know, this is a lethal dose of that bacteria. So this is quite significant. So they published 1940, and this was hugely significant, because look at that date, August 24th, 1940, right? So what is going on in 1940? World War II. And these two guys, as scientists, realized that if you look at statistics around World War I, for example, you know, some of the stats seem to indicate that more people died indirectly from infections than they did from actually bullet wounds, right? And so they said, okay, let's make this stuff. It's going to be hugely important to the war effort. So here's a photograph here of people making penicillin for these guys, or for the government. They didn't want to make large factories. And so, because of course London was being bombed by the Nazis, and so they actually hired people to grow mold in their basement. I find this is one of the most, you know, very ironic kind of historical thing, right? Of people wanting to grow mold in their basement. And they had these special flask designs so they could stack and maximize their space. And this was like, you know, all these, I guess you'd almost call it mom and pop shops of mold growing, and then they would bring it in, and the penicillin would get extracted in a central location. You can see here's an advertisement. Eventually, they brought the mold over to the Americans, and penicillin was mass produced. And there's a lot of unfortunate political crap that goes on around, you know, with the Americans who, you know, patent it and whatnot. But it did help a lot of people in the war efforts. And, you know, afterwards, penicillin was used to treat just about anything, right? Any kind of bacterial infection that was previously incurable. So something like gonorrhea. And a lot that could be said about gonorrhea and various sexually transmitted diseases, but like I said, that kind of the big thing to say is that they were previously untreatable. So this is very, very significant. Okay, so tiny bit more history for you. So you can see we had some successful compounds, and then we had hugely successful compound in penicillin. And we're talking about lots of money being made in these pharmaceutical companies that were previously little pharmaceutical companies, and now we've got massive pharmaceutical companies, and they're thinking, we gotta keep doing this. We want to find things that are, you know, we want to cure tuberculosis. We want to, you know, treat gram-negative infections. So what's the simplest thing to do? You just look for microbes that are making things that might have, you know, a compound that's going to be effective. So the easiest thing to do was just look in the soil. Soil is very diverse. We're talking about maybe 10,000 species in a teaspoon, right? So this is what they did, and this is what people are still doing nowadays. Back then, we're talking about 50s and 60s. They were getting soil from all over the planet. You know, they go to a golf course in Japan, go to the jungles of wherever, and so you basically spread the soil on an agar plate. And you're going to get all these colonies of, you know, who knows what, fungi and bacteria and whatnot. And then what you can do is do an overlay of bacterial culture and look for zones of inhibition. So then what you're going to do, okay, looks like we've got some zones of inhibition here. I'm going to study that bacteria and see what compounds it's producing, and maybe we'll come up with some drugs. There's an example of one there done in, this is from a textbook, a different textbook I have on my shelf. So we're talking about, like, this is a small list, right? Hundreds and hundreds of new antibiotics were discovered in the 50s, 60s, 70s. And you can see streptomyces, for example, are some soil organisms that actually produce quite a few different natural antibiotics. Some come from fungi, and like I said, huge number of drugs. And you may recognize some of these names, and some of them we will be talking about as well. Eventually, scientists, started to bring things to another step. We started making synthetic and semi-synthetic drugs. So what do I mean by synthetic? I mean artificially made drugs. So let's say you have a compound, and you can make something totally artificial, totally synthetic. Many drugs are what we call semi-synthetic. So maybe you take a drug, let's say you take erythromycin, you give it to a chemist and say, play around with this a little bit, and the chemist will maybe add a functional group here or subtract one there, and then we find out that it has it's more potent, or sometimes it's active against different species. And so, like I said, this was done, and now we have hundreds of these antibacterial compounds nowadays. So here's kind of, the timeline, right? You can see most of these drugs were discovered in the 50s, a few in the 60s, and since about 1970, there's a huge gap. There are all sorts of things that were happening. First, all the easy ones were found. Second, there was this belief that, hey, we're good. We got something to treat everything. So the money started to dry up. And third was resistance, which is something we're going to talk about. We're kind of in an era now where, that list you see I have at the bottom, starting around the year 2000 and so on, there's been a lot more research in trying to find new chemical compounds, and we're realizing the need for continued research in these areas. So I'll talk a little bit about resistance in a few minutes. So one of the key points to take away from this historical stuff is this whole idea of selective toxicity. Now, do I have a definition here? I don't. I might have it in your notes. What do I mean by selective toxicity? That is the idea of the magic bullet, right? The idea that you have a chemical compound that is going to be toxic to the pathogen and minimally toxic to the human, right? So hopefully that makes sense, right? We want to kill the bacteria, and we don't want to have any side effects in humans. Now notice I said minimally side effects. Usually there's always something, right? But things like penicillin are often actually very minimal, which is a good thing. Oops, wrong way. So we're going to talk about antibacterials. Like I said, mostly that's all we're going to talk about today. And there are hundreds of these things. And the reason for this is this selective toxicity thing. So if you think about a bacterial cell, it's a prokaryote, right? And a prokaryote is very different from a eukaryotic human cell, right? Think about all the differences, some of the things that we talked about. We have things like peptidoglycan in bacteria, not found in human cells. Bacteria, their chromosomes are entirely different, right? They're circular, they've got different proteins associated with them, their ribosomes are different, and all of these things, believe it or not, are actually targets. So if you can affect, let's say, peptidoglycan, then great, because it's going to kill the bacteria, and the human cells are going to be just fine because we don't have peptidoglycan. Not a problem. You look at that middle bar, fungi, protozoa, parasites, they're all eukaryotes. We have less drugs for them because there are less things about them that are unique than the human cells. And viruses, we even have less antivirals than anything else. And because viruses, they don't have a lot of potential targets. They're mostly using host enzymes. So you have to find something unique to each virus. So it's a little bit more of a difficult kind of challenge to find antivirals. And we really only have, I think there's a couple dozen, maybe 3 dozen antivirals out there compared to the hundreds of antibacterials that we have. And many of them are actually anti-HIV drugs, which have a big market. So more on antivirals probably next day. So let's talk about what these drug targets are. And I have some examples for each of them. I see somebody has a question and says, is Tamiflu an antiviral? Yes, because the flu virus is and we will talk about Tamiflu for sure. Okay, So what are some of these drug targets? So what I have for each of these groups is I have one or two examples. So I know there's a lot of words there. You can see, for example, the cell wall synthesis. There's actually a huge number of drugs that target the cell wall. We're going to be talking about two, and then each of the other categories have got one, maybe two for each of them. So there are some weird words you're going to have to learn. Some of these drugs, as you'll probably learn in your pharmacology class, can have multiple names, which makes it more fun. And these words, like they're weird words, right? So I'll have a comment to make about that when we get to the antivirals in particular. So you can see the cell wall, or the targets include the cell wall, DNA replication, protein synthesis, the membrane, and folic acid metabolism. So I'll cover each of those in a bit of detail. So I'll get to each of them. So like I said, the big one here is the cell wall synthesis, because the cell wall is so unique to the bacteria. They have peptidoglycan, and there's a whole host of enzymes and other things going on there that we can inhibit. And it's a very vulnerable target, right? If you bust holes into somebody's wall, their house is going to fall down. Same thing with the bacteria. Bust holes in their wall, and they're going to burst open. So the biggest group are compounds called penicillins. We're also going to talk about isoniazid, which is an important drug for tuberculosis. So let's talk about the penicillins. So what are the penicillins doing? Penicillins, if you take a look at this, this is kind of a model on how the peptidoglycan is made. So you can think of the peptidoglycan as a big mesh. You've got these, here we go, oops. Time I touch it with my pen, it wants to go. You've got these big, long carbohydrate strands, so there's the glycan part, right? And then in between, we have these peptide cross bridges. So the peptides are kind of joining the strands together and making this, kind of three-dimensional mesh. So what you have here is this group of enzymes that are making all these chemical bonds. Actually, a pretty big group of enzymes. Some of them are called penicillin binding proteins, which is really unfortunate. But these things that's how they were discovered. We didn't know what they did initially. Sometimes in the literature you see them called transglycosylases and transpeptidases, which means something to me as a biochemist, may not mean something to you, but it's at least a better name functionally. So kind of really the bottom line is that penicillin binds these penicillin binding proteins, and they don't do their functions properly. So notice here is we still have the glycan chains being produced, right? But these cross bridges are not made at all. And so now you have holes in the cell wall. And so any growing cell is very susceptible to penicillin, or at least penicillins. I'm going to use it as a plural because there's many different types of penicillin. I'll show you a little video here I have. I thought I had the actual video. I'll have to go to the link there. And you can see there's the terminology, right? Penicillin binding proteins are these enzymes that make new cell wall material, and the penicillins, also known as beta-lactams, I'll come back to that word in a minute. That's just another term for penicillin, lead to holes in the wall. So I'll load up this video here for you, and it's pretty quick. I just have to change my screen sharing. Okay. Hold on a second. Oh, there it is. Okay. So hopefully you can see that, and I'm going to put on to share my volume because there are some sound effects. That's always exciting. Sound effects are cool. So there's a little video here, and okay, let's play it here. Okay, so that was it. I know it's cheesy. You got the popping noises, but maybe you can see some of them are actually bulging just before they break. And that's pretty much it. They try to, as they get longer, they get leaky and they lyse open. So cheesy video, but short and sweet, so kind of funny. Here's my last kind of analogy, right? There's our cell, it's cell wall, right? It stretches out, and you got to put in new building bricks or pieces. And normally that happens, and you get a new cell, and it stretches and divides, and so on, right? Penicillin is kind of like throwing a wrench in there, and stopping construction now, and the whole thing is very fragile and just bursts open, and then there's my cell that's kind of exploding. Okay, so here's penicillin, right? Penicillin G over here on the left. This is the one that was discovered by Alexander Fleming and was purified by Florian Chain. And that was the first one. That was the one used in World War I. It was only injectable, so if you wanted some, you had to take the powder, resuspend it in saline, and get an injection and treat whatever it is, whatever infection it is that you have. You can see there's a bunch of other penicillins here. And notice they're also called beta-lactams. And that's more of a chemist term, and that's referring to that square ring structure in the middle. So hopefully you can see my video here. I have some penicillin. I made this a few years ago. And if you take a look, there is the square ring, right? And so this is kind of the key part of this particular compound that is binding the enzymes that are found in the bacterial species. There's a whole bunch now that have been uncovered. Some of these are, you can see they're natural. Some of them is semi-synthetic, meaning a chemist grabbed it and kind of modified something. You can see the kind of the base part is basically the same for all of them. Notice some of these here, penicillin V, right? That one's super important, and notice, you know, the note beside it, it is acid resistant. So that's huge, right? When we're talking about drugs, if we can have something that is acid resistant, because that way it can survive going through the digestive tract, and it's so much simpler to prescribe somebody, you know, a pill they can just pop rather than, you know, having to get injections and all that all the time. Penicillin V is still prescribed today. It's a really old one. I had pneumonia. I'm trying to think what year that was, a while ago when I was a student. And I remember it very clearly because he prescribed me penicillin V, and I'm like, hey, I know what that is. And, you know, I had my student drug plan at the time. I had to pay the dispensing fee, but not the cost of the drug. And I look at the receipt, and it gives you the breakdown, right? And the dispensing fee was $16, and the drug was $4.25. So I was just amazed that such a cheap drug could have such an amazing effect. So you can see there's some other ones on here. Ampicillin, that's a pretty important one. You still see it prescribed nowadays as well. Also acid resistant, and this one is broad spectrum, meaning it will kill a variety of organisms, such as gram-negatives. And there's a few others down here. The other one that's worth mentioning is methicillin, which is something that is considered a pretty strong antibiotic, and has, again, a decent activity against certain organisms such as Staphylococcus. And you're going to see we're going to talk about Staphylococcus as well. I see there's a couple of questions here. Someone says, so penicillin binds to enzymes that build peptidoglycan. That is correct. Do we need to know the difference between each type of penicillin? The answer for that is no. You need to know what penicillins generally as a category do. And it is useful to know there are different types, but I'm not going to ask you, you know, what's the difference between ampicillin and methicillin, for example. I'm not going to do that at all. Someone's asking, will we be identifying which types of penicillin is needed for case studies? No, again, you need to know what penicillins as a group do. So I could mention that, you know, in a case study, for example, I could tell you that, you know, such and such a drug is used, it's a penicillin. And then maybe I'd ask you some questions about it. How does it work? And so you'd answer that it binds to enzymes that build peptidoglycan or something like that. Okay, so moving on here, that's the penicillins. You can see we keep coming back to penicillin again and again. Very important group of drugs. I do want to mention that there are a number of other ways to mess with the cell wall synthesis. Vancomycin, for example, binds to cell wall components. And it's one of those weird drugs that you used to hear about a long time ago, has all sorts of side effects, and now it's coming back because sometimes it's used to treat infections that can't be treated otherwise because it has such a unique way of doing so. The other group I want to talk about is isoniazid, and this is used for treating tuberculosis. So back to tuberculosis, remember tuberculosis has those unique acid fast cell walls, and we had that mycolic acid stuff. right? So that was that waxy substance found on the surface of the tuberculosis organism. And this is why we have to use the acid-fast stain, because it doesn't gram stain. Nothing permeates that very well. So there is a drug called isoniazid. There's the chemical structure there, and it affects mycolic acid synthesis. And so this is a really important drug for treating tuberculosis. We will talk about tuberculosis kind of later on. I think most of it is we're going to talk about in topic 14 maybe. But I will say this, that treatment of tuberculosis, depending on whether you have drug resistant or not, usually isoniazid is part of that treatment, because there is multi-drug treatment of tuberculosis, but isoniazid is one of the important ones. So there's our, I guess I should have showed that one first, but there's the mycolic acids on the cell wall, making it unique, and meaning that it needs a unique drug. Somebody's asking, do we need to know chemical structures for the midterm? Nope, this is not a chemistry or biochemistry class. I show chemical structures because I am a biochemist, and I like chemical structures. And sometimes it helps me remember what I'm looking at. Sometimes in the names of the drugs, there's clues as to what the structure is. If those things help you, great. If chemical structures don't help you, just ignore them and learn the names. Okay, let's talk about drugs that damage membranes. So there's a few of these. I just want to talk about one of these drugs. Actually, there's a bunch of them, but the polymyxins. There's a few of these polymyxins. Oops, polymyxins. And if you remember that gram negatives have a unique outer membrane, and so that's what polymyxins target. So again, we've got some sort of structure that is unique to the bacteria and not found in the human cells. So this is great. These things are relatively toxic, and so they're usually used on topical things. So there's a picture of polymyxin B, and if you want to learn a chemical structure, that's the one. That's a really, these structures, they fascinate me. I mean, if you show this structure to an organic chemist and ask him, can you synthesize this? He would say, not in 100 years. I mean, that thing is super complex. And yet the organism that made it, didn't have a problem making it. Some organisms are very good chemists, very fascinating molecules. So remember, the outer membranes are unique. They have some unique lipids called lipopolysaccharides, and the polymyxins kind of get in there. They disrupt the whole thing. And again, you're disrupting the cell surface on a bacteria, and that's going to weaken it, and it can die. So as I mentioned, these things are often used kind of topically. So maybe you're thinking poly, where have I seen that word before? Poly something and something topical. You're probably thinking of polysporin. So I don't know what your mothers are like, but my mother was obsessed with polysporin. And any little scratch I had as a kid that required a Band-Aid, she would stick that polysporin on to kind of prevent infection. And so there's a whole bunch of these out there nowadays. You can see polysporin. The basic brand has polymyxin B in it, and another antibiotic there as well. And there's, like I said, there's a whole bunch of these out there. I was in the pharmacy and just kind of taking a look at some of these. There's some there's like the Shoppers Drug Mart brand, I can't remember what it is, Life brand or something, which was exactly the same as polysporin. And then I saw Neosporin, and Neosporin had 3 antibiotics in it. So I don't know if that's that much amazing and better, but I saw that, and then I looked, and on the shelf there was another polysporin. I can't remember, it had a different color, and it had the same 3 in it. So and then I'm like, well, way to confuse all the shoppers, because I don't know which one to buy now. But the whole idea is, like I said, it's a topical cream, and it's going to have antibiotic properties, and so you can use it or put it on your kids. And I know my mom was a big believer in it, and I've certainly had a scratch or two that did get infected, and maybe I should have used a little bit more. Okay, category 3, drugs that inhibit protein synthesis. So remember, protein synthesis happens at the ribosome. So there's our ribosomes, and you can see they're grabbing onto some messenger RNA, and they're reading the genetic code and they're putting together amino acid into a string, and that's making a protein. So also remember that prokaryotic ribosomes were 70S and eukaryotic ribosomes were 80S. So what did that mean? It means that they're different sizes and they have different components. If you look at the components, they're made of RNA and proteins, and they're different. Different enough that these drugs will preferentially bind a bacterial ribosome and not bind a eukaryotic ribosome. So this is great, or at least they don't do entirely. Some of these do have side effects because they will, in high concentrations, affect eukaryotic ribosomes. But so this is why you have all the testing, because some are really nasty and some actually do a pretty good job. So there's actually a couple here I want to share with you. This one here, erythromycin. Again, we've got a really fantastic chemical structure that would be very hard to synthesize by a chemist, but a bacteria has done it. I think a Streptomyces is from. And these bind the large subunit. So notice on the ribosome there's two subunits, right? And this one blocks some function in the large subunit. So erythromycin, very important drug. I think I have a note. No, I don't have a note there. Maybe I have a note in your notes, but I just don't have it on the slide. That this drug here is the one that if you have a penicillin allergy, right? You have a penicillin allergy, so you can't give. There's like 100 different types of penicillins. And somebody has an allergy. How frustrating is that? We can't give them any of those penicillins. So this is kind of one of the #1 choices is erythromycin. You give someone erythromycin because it's totally different. Has a totally different chemical structure. Works in a totally different way. Erythromycin is also effective against a lot of gram-negative infections. So I think my understanding is it's pretty commonly prescribed when people have bladder infections. So bladder infections are often caused by E. coli, and erythromycin is an option for that. So a very important drug, erythromycin, and there are a few derivatives as well. I'm not sure whether they're all called erythromycins, like some of them's A, B, and C. It's kind of hard to keep track. There's so many of these drugs out there, and then they have common names and they have brand names, so somebody is saying that's the name brand of erythromycin. Yeah, so sometimes it gets a little confusing with that. Tetracycline, another huge important brand of antibiotics, and they bind the other part. They bind the small subunit. So if you want to look up the exact mechanism of action, that's fine. I'm not going to say anything more than that, but you can see it says here they block transfer RNA docking, if that means something to you. So tetracyclines are usually considered, again, sometimes people talk about antibiotics being strong. Tetracycline falls into the strong category, meaning that there are some organisms that nothing else works, right? But tetracycline will kill them. It has a really good activity against gram-negatives as well. Some gram-negatives like Pseudomonas aeruginosa, we talked about it, we mentioned a couple times. I think I mentioned that it's one of those organisms that's kind of naturally resistant to all sorts of things. The tetracycline will do it, but higher concentrations than if you're trying to kill E. coli, unfortunately. So one thing that's interesting with tetracycline, I've worked with tetracycline in the lab, and the chemical itself, it comes as yellow powder. And I didn't know this, but you don't prescribe tetracycline to children because of that yellow color. For some reason, I don't know all the physiology behind this, but it'll actually attach to growing bones. And so if children get tetracycline when their teeth are growing, the yellow color can actually end up on the teeth. And I Googled this, and apparently there's a bunch of dentists that are willing to, they've got some treatments that are going to help you with that if that is something that happened to you. But a lot of people, somebody is saying, what about skeletal bones? Probably, but we don't see the skeletal bones, and so, not really an issue. I think mostly it's the discoloration that we don't like. I don't know if there's any other necessarily side effects. I know people that have had skin infections as well. I knew somebody who had some extreme acne, and they were on a long course treatment of tetracycline, and I don't really know what the result of that was. I just know I was concerned because they were supposed to take tetracycline for weeks, and you know, I had concerns about that, what that's going to do to all the other microbes in their body, because this is a very strong drug. So one other I want to mention, just for historical reasons, streptomycin was the first treatment for tuberculosis. It also binds the small subunit. So it's one of those ones that, you know, there was no treatment, and then we came up with streptomycin. And you can imagine the difference this made historically when tuberculosis was at one point, you know, widely prevalent disease. So like a lot of these antibiotics, and the other reason why I bring up streptomycin is, you know, they have side effects, particularly these things where you're binding ribosomes, and, you know, eukaryotic ribosomes, they can bind there as well. And I don't know all of the physiological reasons why this, but streptomycin and some of these other ones are known to cause ear damage. And so presumably has something to do with the ribosomes, and that the drug is not perfectly safe, but a good historical reason why we don't use it anymore. Yes, this title should say protein synthesis. There was a mistake there. I probably just copied a slide. Did I make a mistake on the previous slides? That's one good question. I did. Look at that, hey? Okay, somebody with sharp eyeballs. Good job. Yeah, so I'll have to remember to fix that when I get back to my office. Okay, so two more groups worth mentioning. Drugs that inhibit DNA synthesis. So it turns out that, thought I had a nice slide here, yeah, so I'll go back to that in a second. Turns out that the enzymes that do DNA replication in bacteria are different enough from the human versions that there are some drugs that can get in there and affect DNA synthesis. The fluoroquinolones are really important. This is one of these ones that my understanding is they've been around for a long time, but were not actually used except for in the last 20 years because, you know, it's something that they discovered a long time ago, but hey, we had penicillin, so why, you know, investigate this? But, you know, with all the resistance nowadays, these things have popped up a lot more often. So fluoroquinolones, you can see fluoro, that stands literally for fluorine, the element is found on these. Sometimes you see people will just say quinolones, but there's another drug that's kind of, there's the quinines as well, so it's nice to put the word fluoro in there to distinguish the difference. So here's Cipro, or ciprofloxacin. This one only came out, I think it only came out in 2001. I know people are thinking, well, I was born in 2001, a couple people here. But you know, historically for antibiotics, that's not long ago. It's at least within my memory of remember when it came out, and it was a huge game changer because there was no resistance against it, at least initially. So how does this thing work? Like I said, there's a bunch of enzymes involved in DNA replication, a whole bunch. And some of these enzymes here, you can see have two enzymes here, topoisomerase and DNA gyrase. And so when DNA is being replicated, you've got this double helix, and it's a helix, right? So it's kind of like having a hair braid, right? You don't just yank at it and pull it apart because it's going to knot up. You know, you take it apart carefully. And this is actually what these enzymes are doing. They're kind of helping the DNA to come apart carefully and untangle itself. So if it gets tangled, the DNA just gets stuck, right? And then the organism can't grow. So this is what the quinolones do. They kind of get in there and they affect these untangling enzymes, and then the DNA gets tangled, and that ultimately kills the organism. So that's kind of what they do. And these enzymes, like I said, they have these names here. You can remember those names of those enzymes, or you can just remember the concept, right? That they're involved in that aspect of DNA replication. Okay, so one more group, and this is kind of a miscellaneous group because there are actually a few that do a few other things. There are a few pathways that some bacteria have that we don't have. And a big one, and we've actually already talked about this drug, these sulfonamides, inhibit folic acid metabolism. So you're probably thinking about folic acid, and you know, what is that? It's one of the B vitamins, right? And so that's great. We like B vitamins, but we can't make them, right? That's why B vitamins are a vitamin, because our bodies can't synthesize them. We have to take them in our diet, right? Turns out that a lot of bacteria don't take them in their diet, they make them. So this is kind of showing a chemical pathway, right? You got some sort of precursor, you get this intermediate called PABA, and you got some more enzymes. Eventually you get folic acid, which eventually gets made into nucleotides. So it's an essential thing, essential for us to make our nucleotides, but we can get folic acid in our diet. A lot of bacteria, they don't get it in their diet, they synthesize it. So these sulfa drugs, sulfonamide, this is Prontosil, which I mentioned as a historical example. This is what the drug looks like. So notice it's kind of an interesting chemical structure. It's very similar to that PABA thing, right? So that, and so it gets in the enzymes, and basically no folic acid is made, and then they can't make the nucleotides. So you're targeting something that's unique to the bacteria, is really kind of the bottom line here. I'm just going to take a look. I think I have a couple of questions here. Somebody's saying, does streptomycin inhibit DNA or protein synthesis? Protein synthesis. It was a mistake on the heading of the slide, but if you look at the picture on the slide, it is affecting the ribosome. Are sulfonamides commonly prescribed? That's a good question, and I don't exactly know. I know they were prescribed decades ago, and then penicillin came out, and people stopped prescribing them. My understanding is they are starting to get some more, they're starting to get prescribed a bit more for some cases of resistance. My understanding is that some of these sulfa drugs actually have anti-parasite activities as well. So I think there are cases where people have certain parasitic infections, which is why sometimes they get prescribed. I see a couple of people have some other comments, and somebody has a sulfa allergy. That's not good. And somebody says in their daughter's case, they were prescribed after the other antibiotics didn't work. So that confirms what I heard. But like I said, I'm not as familiar with this particular group as some of the other ones. I just know it for historical reasons. So there's kind of the list, right? The different drug targets, and those are the examples you should know. So, you know, lots of words and stuff, and I'm looking at 1, 2, 3, 4, 5, 6, 7. So seven drugs to know. That's not too bad. I mean, we still have the antivirals, and we still have some of the other groups to know. I'm sorry, but we're not going to hopefully cover those today. Somebody just asking questions. So folic acid is needed by bacteria to make their DNA. Yes, basically yes. And they don't get it in their diet. They synthesize it. So for us, it's also essential for us to make our DNA, but we can get it in our diet. The bacteria, they make it synthetically, and they don't get it in their diet. So just kind of a different route, and it's a vulnerability on their part. Okay, so I kind of want to talk about some different aspects of antibacterials, and this is what I was hoping to cover today, which is good. So some definitions. I know I've dropped these words a couple of times, and somebody asked a question about one previously as well. Some drugs are considered broad spectrum, and some are considered narrow spectrum. So what do we mean by that? Broad spectrum means it can kill a whole bunch of things, both gram-positives and gram-negatives, and narrow spectrum means it might have more limited activity. So I kind of mentioned some drugs in those categories, right? Like a lot of the original penicillins were pretty narrow spectrum for gram-positives only. There's some more broad spectrum ones now, such as ampicillin, which is a lot more broad spectrum. Some of these broad spectrum drugs, these fluoroquinolines, so such as Cipro, and these tetracyclines are broad spectrum. That's why often people use the word powerful, meaning you can kill lots of things. You know, I don't really like to use the word powerful, you know, or strong necessarily all the time when we're talking about chemicals, because you know, what does that mean, right? I've taken enough chemistry classes to know it's just all a relative thing, right? When, you know, something might be strong compared to this one, but then you can always find something stronger. So I like to use, you know, broad spectrum and narrow spectrum are better terms, and they're more precise. So somebody's asking what type is preferred. Well, it turns out, that's my next slide. advantages and disadvantages of broad spectrum antibiotics. So some of these are probably obvious. You know, why would you use one over another? So broad spectrum, right? You don't know what the infection is, or you're suspecting Pseudomonas, or you're suspecting resistance. You know, it's very easy to prescribe a broad spectrum antibiotic, and that's great, because then the patient, you know, they get their prescription and then you never see them again, right? And that's a good thing. What are the disadvantages, right? Well, first of all, you know, and I don't have it on this list here, but, you know, from my perspective as a scientist, my biggest thought with broad spectrum antibiotics is that we want them to be effective. We want them to be effective in the future. I don't want resistance to evolve against broad spectrum antibiotics. And that is the case with Cipro. Like I said, I remember it coming out as a case study as a brand new drug, and how fast it would take for resistance to pop up. And I think it was 2 years. So suddenly you had a drug that was effective for sure, you know, and then, you know, you're kind of like, well, is anything a sure thing? It turns out, no, Cipro is not a sure thing, because eventually resistance did start popping up. Another disadvantage is broad spectrum antibiotics are very good at messing around with your normal flora. So with your intestinal bacteria and other things. So something I think that I talked about before is sometimes, you know, people get antibiotics and it causes other organisms to flourish. So we talked about last day, we talked about Clostridium difficile. So Clostridium difficile is difficult to kill. Not all antibiotics will kill it. And so, you know, you have some sort of infection, and if you're colonized with Clostridium difficile, you know, the antibiotics don't kill the endospores. So then it has a chance to flourish, and you're getting what is often referred to as antibiotic-induced diarrhea. Another case where broad spectrum antibiotics can cause issues is women and yeast infections. So think about it, yeast is not a bacteria, right? So maybe a woman gets antibiotics for something else, could be pneumonia, could be strep throat, who knows? They get prescribed broad spectrum antibiotics, and it, you know, it just really does a number on their normal healthy bacteria that are normally in the gut, and in this particular case in the vagina, and now the yeast have a chance to flourish. And so this is a pretty common occurrence in some women, because the yeast is just part of the normal flora, but once it gets that chance to kind of flourish, because the antibiotics wipe everything out, that's the case why it causes yeast infections. I see there's some comments and questions. So somebody's asking, can it make people more prone to contracting C. diff by taking antibiotics? Yeah, so it makes it easier for a person to colonize as well if they're on antibiotics. Somebody's making a comment about people eating yogurt while taking antibiotics. And yes, we're going to talk about probiotics a couple of units from now. Yogurt is considered a probiotic. And there's some good studies to show that taking yogurt while taking antibiotics does help some women. Unfortunately, everyone's different, right? Some people it seems to help, and some people it seems to not. And you know, this is where some of the debate and all that around yogurt and antibiotics comes in. So a bit more for a later lecture, but good question. So some other definitions for you. I'm not too concerned about going into these details on these drugs. This is something that might come up in your pharmacology class, but some of these drugs are bacteriostatic and some are bactericidal. So kind of good words to know, and like I said, you might be going over this in more detail in your pharmacology class. But what does this mean? It means these static drugs don't necessarily kill them, but they kind of, you know, slow down their growth, right? And that's usually enough sometimes to get rid of the disease symptoms, or enough for your immune system to finally come in and kind of, you know, do the rest of the job. Bactericidal means it's going to actually kill them, right? So, you know, sometimes it depends on their mechanism of action. Like I said, both have their place, and we just don't have time to kind of get into all the nuances of this in this course, but other than to throw those words at you as something to know. Okay, so let's talk about testing antibiotics. We kind of talked about testing disinfectants, and you can do some of the same tests for testing antibiotics. So we can do this disc diffusion assay. also called the Kirby-Bauer test. And you can see it's the same idea with the disinfectants. What you get is a little disc, a little filter disc. The antibiotic is in the disc. It gets put onto a lawn of bacteria, and you can look at the zone of inhibition. So if you take a look at this, we've got, you can actually buy these things. So we've got AM is ampicillin, K is kanamycin, TE is tetracycline. Notice it's kind of got a little bit of a yellowish color. We've got PB for penicillin B, and the concentrations are written on the discs. GM is gentamicin, so that's probably micrograms per mil or something like that. And you can see that tetracycline and kanamycin are the most effective. They have the biggest zones of inhibition. So this is a super easy test to do, done in clinics all the time. Someone has, they need to do some drug susceptibility testing on a patient because the treatment's not working. You plate it onto an agar dish and you pop it on a bunch of different antibiotics and you see which ones work. So very, very easy to do. Doesn't necessarily give you all the numbers, right? In this case, we can see that, gentamicin is fine at 10 micrograms per mil. Well, what about 5? Like, we don't want to over-medicate people either. So you can do more kind of particular tests. So if you remember with the disinfectants, we can make dilutions. You can do the same thing with antibiotics. This is called an MIC test, or a minimal inhibitory concentration test. I've done these as well. And you can see, like, what we have here is we've got tetracycline again, and you've got the concentration written on the test tube. So you've got 0.2 micrograms per mil, 0.4, 0.8, 1.6, 3.1, 6.3, 12.5, and so on. So they've just doubled it or halved it, depending on how they end up making the dilutions. And you can see very clearly right there at 1.6 micrograms per mil. That's the minimum concentration required to inhibit the growth of this organism. So this gives you some exact numbers, and it can be done to look at, let's say you have a new strain, and we want to know the drugs doesn't seem to be working, but what if we use a higher concentration? In some cases, that's the case, right? Just different strains or even different organisms, have different vulnerabilities to the drugs. So is MIC used to not over-medicate the patient? I'm not sure if it's used necessarily in clinical labs. I would have to check into that. It's more used for when pharmaceutical companies are trying to figure out which drug to use in which case and what to recommend to doctors. My understanding is used in some rare cases where people are, desperate and trying to figure out what to do, but usually the standard thing is, okay, erythromycin is not working, let's just try tetracycline, right? That's usually the standard way to do. You try a different class of drugs. And unless it's a complicated case in a large university hospital, I'm guessing it's not a standard kind of thing to do. So what about combining these tests together? So we have the E-test as well. Okay, this is kind of cool. I've never done one of these. I didn't even know they existed until about 10 years ago. You can buy these strips. And rather than buying a little puck or a little disc of one antibiotic concentration, you can buy these strips that have a range on there. And you just throw them on the lawn, and you can read directly off there. Maybe a little less precise, but useful information and super easy to do. So back to this slide here, right? We have this innovation gap and we have these new drugs. So what is going on? So we need to spend a little bit of time and we need to talk about resistance. So I don't know why my slide had a beautiful animation for you. It's not working. Okay, oh well. Go to this slide. This is a beautiful picture. I have to substitute for the beautiful animation. What I was saying in the previous slide is that resistance, bacteria are striking back, right? And so what is going on? Well, you know what? These things have been living out in the environment, in the soil, on trees, and there are, possibly 10,000 species in a scoop of fertile soil. So they have to carve out their own niche. And literally they are waging chemical warfare against one another, trying to kill each other and trying to live. And so it shouldn't be surprising that they have mechanisms to resist various compounds. And so these things, like I said, this has been happening for a long time. And ever since penicillin was marketed in World War II, I think even before the 1940s were done, people were starting to document resistance. There's all sorts of studies like this. You can see this is looking at resistance in gonorrhea between 1980 and 1990. And it doesn't say what it is. I got this out of a textbook. You can see going from about 0 to 8 or 9% of cases. Probably, I'm guessing this is for penicillin V or something like that, an older drug. But lots of studies showing this kind of thing, that resistance is happening and it is on the increase. In some cases, some very scary studies. So this is a real concern, right, that our drugs are not effective. Let's talk a little bit about how these things work. How does the bacteria resist? There's a bunch of, I guess you'd call them cell biology kind of mechanisms. And I'm just going to sort of, got this little graphic here to show you what's going on, kind of from a basic level. First thing is the drug can be destroyed. So that organism has obtained an enzyme that can destroy the drug. Another mechanism is the drug can get altered, not necessarily destroyed, but maybe, the drug is, a functional added onto it so that it's too bulky to fit in the enzyme or something like that. It's usually considered a different mechanism by all the scientific literature, so that's why I've included it. Another mechanism are these efflux pumps. What does that mean? It means pumping drugs out. It may seem kind of weird and it costs a lot of energy, but it works. There's a lot of organisms that have very good pumps. Why they have them? There's a number of reasons, right? They're encountering environmental toxins all the time. So if they see something they don't recognize, it just gets ejected. And these things are really effective against very high concentrations of antibiotics for some organisms. So very effective mechanism. You don't need to remember this chart here, but I'm just showing you for those drugs that we talked about, you're looking at all of these types of resistant mechanisms. You can see, for example, just looking at ampicillin, there are mechanisms, all three types of mechanisms are used by bacteria to resist ampicillin. So this is quite something. Cartoon for you, you can see it says here, it was on a shortcut through the hospital kitchens that Albert was first approached by a member of the antibiotic resistance. And the guy says, "Psst, hey kid, want to be a superbug? Stick some of this into your genome." So I kind of want to talk about how this happens, right? How do you get enzymes to do this? Well, you need new DNA. And there's actually 2 slides I have to share with you for this, how they acquire resistance. One is called vertical transmission. What does vertical transmission mean? Usually it means, mother to child or something like that. So you can think of it as that, right? Going from one generation to the next. So this is just really selection, okay? I'm not going to call it natural selection, okay? Because, we're using drugs and stuff. This isn't happening naturally out in the wild, but it is a form of selection. And so you've got this organism growing on you or in you, and they grow and they sometimes have mutations, right? And it's pretty rare. But if you think about like these organisms, right? They're doubling, and you can have an infection, and let's say it's a digestive tract infection, and so you've got millions and millions of bacteria. So if the mutations are rare, it's still possible you might get one in something that is a little bit less sensitive to the drug, right? And so all it takes is, and then somebody takes their prescription, and they get, and that little guy, you can see he survives, and then has a chance to grow and propagate. So this is kind of one of these reasons why if you ever get antibiotics and somebody, the doctor is going to say, and make sure you take all the pills for the full 10 days, even if you feel better, right? The whole idea is we don't want people to have kind of low concentrations of the drug in their blood and then, promote the selection against these, for these resistant organisms. We want to have the high full concentration. We want to make sure that all of them are cleared and we're not getting any resistance. So this is one way that can happen. There is something called horizontal transmission, which in some ways is even a little bit scarier. So let's take a look at horizontal transmission. You can see there's my organisms like before, and it says horizontal transmission is obtaining DNA from other sources. And how can you get DNA from other sources? One way we talked about already was bacterial conjugation, right? That is bacterial sex, so they're exchanging DNA, maybe not even the same species. There are other mechanisms. We can also have transformation. So gonorrhea, for example, is very good at this. Good at what? Good at just scooping up DNA from the environment and trying it out. And if it has useful genes, it'll keep them for himself. Another way is something called transduction. Probably a little bit more rare, but viruses can actually move DNA around between cells. So in this case here, what you have is not just kind of subtle and slow mutations, but you have these organisms that are getting brand new genes, and boom, just overnight, suddenly they have resistance to drugs. So you can see I've given them little nasty eyes and horns and things like that. And sometimes they can actually obtain resistance to several drugs. So I can just see I'm almost running out of time, but I will share this video here for you. I'm just going to make sure that the volume is on. It's a short video, and this is an experiment done. And it's a good thing it's not nighttime, because this is terrifying. This is one of the scariest things I think I've ever seen. So I guess just in time for Halloween, right? And you can see what they're doing is they're doing an experiment with a giant petri dish. So I'm going to press play, and he's going to explain what's going on there, and you can take a look at this video.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Okay, so I know everyone is having a chill down their spine now. Yeah, this is terrifying stuff and pretty crazy. So this is what we're dealing with, right? We have bacteria that can find resistance. Now, he's not saying everything about this study in terms of what drug he's using. It's something that's very easy for organisms to be resistant to and all that. But you get the idea, the principle behind it, and the visual aspect of this, and going from to 1,000 times what E. coli normally can resist in a short amount of time. I think this whole thing was actually a couple of weeks. So we're going to talk a little bit more about resistance when we get back next day. We want to talk a little bit about superbugs, and then we'll get into those antivirals and antifungals and those kind of things. So anyway, that's the end. So hopefully everybody has a great day. Do try to sleep tonight knowing that these things are out there, and we'll see you on Thursday.